Thank you very much for allowing us to share our work. So in this talk, what I'm going to do is to tell you what we are doing in terms of Lumella test, which is essentially glycosylated fibronectin for the assessment of preeclampsia. Now, right from my medical school times, we were told that pathophysiology of preeclampsia is quite well understood, although the downstreams are not so well understood. But there are two key factors there: vasospasm and endothelial dysfunction. And traditionally, vasospasm has been assessed by high blood pressure and endothelial dysfunction leading to proteinuria. And these two symptoms, if you like, they have been used for diagnosis of preeclampsia. And for years, preeclampsia was based on association of high blood pressure and proteinuria. But over the last few years, we've realized that there are biomarkers which are also important, elevated, and useful for assessment of preeclampsia. And this slide I've lifted from the work of Sanen, and that's been published a few years ago, 15 years ago, actually. And he's looking at the performance of several biomarkers for diagnosis of preeclampsia. Any diagnostic test, the parameter that we're looking for is the area under the receiver operating characteristic curve, and higher the area, the better the test. And you can see that one of the tests which already is in clinical use is VEGF risk factor receptor 1, and that is the S-FLIT. And he reported the area under the curve was 0.91. And very similar area under the curve was also reported for this biomarker called fibronectin. Now, fibronectin is a protein which is present in the serum. It's also present in non-pregnant individuals, and it has got several isotypes. And a particular variety of fibronectin called the glycosylated fibronectin was particularly associated to be with preeclampsia, and that is where the whole work was concentrated on. And following the laboratory work, looking at the particular protein, the test was then made into commercial product. Now, several studies using the glycosylated fibronectin have been reported, and we are at the minute looking at did a systematic review about the sensitivity and specificity of glycosylated fibronectin for the diagnosis of preeclampsia. And in this particular manuscript, sensitivity of 0.81 was reported by several studies, and a specificity of 0.85 has been reported. And this is a pooled specificity that we are talking about. So there is interest, there is promise in the use of glycosylated fibronectin. Now, following that, we embarked upon a study of comparison of glycosylated fibronectin versus the established S-FLIT-PLGF ratio for the assessment of preeclampsia. And we were kindly funded by the National Institute of Health Research in the UK. And they wanted us to look at a head-to-head comparison with these tests. They also wanted us to do a health economic analysis to make sure or to assess whether it is going to be useful as far as the money goes for the NHS. So this is a prospective multi-center study, and women at risk for preeclampsia based on predefined clinical or obstetric risk factors will be invited for the study, and the recruitment will take place at gestational age between 24 plus 0 and 26 plus 6 weeks of gestation. Now, this is a test which will perform the point of care testing for glycosylated fibronectin. The beauty of this is that this test will be blinded. The results of the Lumella test will be blinded to the clinical team. So clinical team will not know what the results are, but they will know the results of S-FLIT-PLGF ratio, which is on the Roche platform. And now NIHR also wanted us to do a Quidel test, which is essentially PLGF testing, but that we will be doing on stored samples. So women turning up with suspected preeclampsia will undergo the SLED-PLGF ratio as it happens in the routine clinical practice, but they'll also have a point of care testing with the Lumella test. And this has been funded by NIHR, as I explained to you. So this is the photograph of the kit that is used here. So you can see that there's a cassette on which blood sample goes, and that is a finger stick sample which goes onto the cassette. The cassette goes into the reader, and the reader will give us a result, a quantitative result as to what the levels of glycosylated fibronectin are in about 10 minutes time. So this is really a point of care test. It does not require venipuncture. It does not require anything else to be done to the venous to the blood sample, and it can give us results without the sample having to be sent to the laboratory. The hypothesis is that this will be detecting the primary outcome is delivery for confirmed preeclampsia within 4 weeks of testing. And the hypothesis is a non-inferiority. We are testing it as non-inferior to the currently used SLED-PLGF ratio. It's a non-inferiority of point of care test compared to the SLED-PLGF. The sensitivity is the primary endpoint. So we argued that one of the most important parameters is to be able to exclude the diagnosis. When women come up, can we confidently say to them that, look, you're not likely to develop preeclampsia in the near future, so you can safely go home? And that's where sensitivity is important. And we set the non-inferiority margin of 5%. The plan is to recruit a total of 400 participants over 18 months period, and the recruitment is already started. The recruitment will take place from 3 sites at the minute: St. George's, St. Heliers Hospital, and Epsom General Hospital. And we expect to complete that recruitment of 400 participants in about 18 months period. We anticipate the prevalence of preeclampsia to be about 23%, and that figure comes from the INSPIRE study, the study reported by Manu Atish and his Oxford colleagues. And in their study, the prevalence of preeclampsia was 23%. So that is what we have used for calculation of the sample size. And the results of the Lumella test will be dichotomized into low risk and high risk based on study-derived thresholds. So we haven't got, because this test is going to be done as a blinded test, this will not be used for clinical decision making at the minute. What we do want to find out is what is the best threshold that we can use when we test it head to head with the SLED-PLGF. Time to development of preeclampsia and delivery for subjects will be assessed using Kaplan-Meier curves and Cox proportionality hazard models, and that is the statistician's job. The study has been given ethical approval by the London Harrow Research Ethics Committee in last year, in October, and we've started recruitment on that, and the study is registered at a publicly available registry as well. What about the study team? So the study team is AG Health, that represent Diabetomics in the United Kingdom, and Diabetomics are the company which developed the test in the first place. They are the ones who they developed the test, they made it into a commercial product, and we are working very closely with them because this study was funded through the Research for Innovation funding stream of NIHR. It is the industry which are the primary grant holders, and we are working with them as a collaborator. The other one, the study team, is what is now called GESH NHS Trust. Now that we have merged George's, Epsom, St. Heliers, they've merged as a single trust. So we are 3 hospitals there. All the samples from the study go to Kingston Pathology, and Kingston Pathology Lab is on board. That's where the samples are received. SLED-PLGF testing is done. Results are disseminated and given, and the samples are stored there. But obviously, point of care testing happens at the site where recruitment happens. Kingston University is where our statistician is based. York Health Economic Consortium is also involved because they will be doing the health economic analysis. And we've got two teams, the KT's team, which is based in Queen Mary University, and they are a patient public initiative engagement team. And of course, we are closely associated or involved with APEC, because APEC was also our co-applicant for the funding application. Now, when we, or any of you, come to St. George's, Epsom, or St. Heliers Hospital, this is what you will see displayed in the day assessment unit and antenatal clinic, giving information to potential participants about the presence of the study. And we are very excited that we have already started recruitment. What benefits are we expecting out of this? So this will enable the test to be done by midwives used during patient visits. So midwives themselves can do this test and obtain a result. It'll provide a quick answer to an important question. It will identify women at risk of developing morbidity sooner, and it will avoid delay in diagnosis. It will also, we expect significant savings per patient in the NHS by correctly identifying patients who will develop and identifying patients who can be reassured that they are at a low chance of developing preeclampsia. NIHR was very particular that they wanted us to do a head-to-head comparison with the existing test which is present in clinical practice. Now, the existing test requires venipuncture. Blood sample goes from my hospital, that is St. George's Hospital, goes to Kingston Pathology, and the test results sometimes take up to 24 hours to come back. So they are not available straight away. It is a pragmatic real-life study. Many of the test results that I showed you earlier on point of care testing, they are done on stored samples. They are not done on patients with a finger stick sample. So this is a real life study. There is a health economic analysis also involved with it, and there are huge benefit for sites with limited laboratory access. Just imagine somebody who's working in the field where there aren't any labs, but if they've got access to this point of care technology, then it is possible to identify women who are likely to develop or at a high risk of developing preeclampsia. So we are sitting at about 150 as far as recruitment number is concerned. The study goes on till next year, and I don't have the results, final results by as of now, but I wanted to tell you what we are doing with APEC and AG Health regarding the Lumella test, and I'm sure that I will be able to stand here in time to come and tell you what the results of the study are. Thank you very much for giving me a patient hearing. Thank you.